Okay, so here we're going to talk about mitotic cell division. And this is the typical cell division that built your human body. Mitosis is the word we use to describe the process of cell division. And the most critical event in mitosis is division of the nucleus. Um, in recent years, mitosis is also used to include the following division of the cytoplasm after a nucleus divides. Some biologists still separate it, but I think in our unit here, we're going to go with mitosis including cytokinesis. So here's how it works. A cell that's not in the process of cell division won't have any chromosomes. This cell right here, the way that this cell is drawn, it appears to have some chromosomes. So it is not a cell that's in interphase. So cells that are in G1, S, or G2 will not have chromosomes in them. They will look something just like that. A nuclear membrane, a nucleus, a little nucleolus inside, and then organelles around. This cell doesn't look like that. Its nucleus actually has X's in it. This cell is what we say in prophase. Prophase is the first phase of mitotic cell division. It takes usually the longest of all the phases, but not always. In human cells, metaphase is usually longer, but it just depends on the cell type. During prophase, the chromosomes become visible because the DNA supercoils up. In addition, the nuclear membrane dissolves away. And then the centrioles divide and they pull microtubule spindles between them. And that's what you see fully formed here. There are spindle fibers radiating out from the centrioles across the cell. Now, in prometaphase, the spindle fibers complete formation and each chromosome attaches to one of those spindle fibers. Okay? So the difference between prophase and prometaphase is in prophase, the centrioles divide and start separating, the nuclear membrane starts to dissolve, the DNA starts to supercoil. In prometaphase, which is usually pretty short, the chromosomes attach to those spindle fibers at the centromere, and the nuclear membrane finishes dissolving. Now, the next phase is called metaphase, where the chromosomes line up right down the center of the cell, but they're not single file, it's three-dimensional. They line up on a dinner plate surface, on a circular region in the middle of the cell. And the reason is the spindles push the chromosomes there using their centromeres. Now, for a human cell, that takes a long time. And then there's anaphase, where due to activity at the centromeres and a calcium release, the centromeres break and the chromosomes actually literally walk down the spindle fibers in opposite directions. So this little chromatid walks opposite to this chromatid, and this chromatid separates from that chromatid. They were together as identical pieces of DNA from DNA replication, but now during anaphase, they're being separated. And then during telophase, the DNA that's separated at opposite ends reforms a new nucleus around itself here, another nucleus, another nuclear membrane around itself there, and then the cell pinches in half. That's the cytokinesis part, and then you end up with two cells separate. So here's what we've got. This is prophase, this is prometaphase, this is metaphase, this is anaphase, this is telophase, and then this is cytokinesis. Here it is animated. Let's look at the animal cell mitosis first. That's a G1S or G2 cell right there. You can't tell, which phase it's in. I didn't draw any other crazy organelles. I just drew everything as dots outside the nucleus. It looks like an egg in a frying pan, but this is the nucleus. That's a nucleolus, and this is the cytoplasm here, and this is the cell membrane going around. Now, here we begin prophase. The DNA supercoils. You'll notice the nucleolus disappears, the nuclear membrane disappears, and at this point, we're in prometaphase. I didn't distinguish that, but this is prometaphase, where the centromeres actually attach to the spindle fibers. And so now what we've got, the nuclear membrane's gone, the nucleolus is gone, the spindle is formed, the centrioles have gone to opposite sides, blah, blah, blah. Now we have metaphase, where the chromosomes are pushed to the center of the cell. Okay, now that's metaphase right there. And at this point now, calcium is released, which causes it causes an enzyme to split the centromeres. At that point, the centromeres split. Now, this is an identical piece of DNA to this. This one's identical to this one. They're semi-conservative replicated copies of each other from DNA replication. These two are identical. These two are identical. These two are identical, and so on and so forth. So when the cell separates them, this wad of DNA here, this 1, 2, 3, 4, 5, 6, 7, 8 pieces of DNA are identical to these 8 pieces of DNA. What we have here is a cloning. So the chromosomes are pulled to opposite ends. That's anaphase. And then we have telophase, where the nuclear membranes form around the two separate nuclei, and then we have a cytokinesis. The cells pinch in half. And that's mitosis. That's all it is. I don't stress really the names of the phases anymore. You just need to know what's happening. So to reset it, we have chromosomes supercoil, chromosomes attached to spindle fibers, chromosomes line up. Calcium release causes centromeres to snap. Chromosomes that are identical separate. Anaphase moves the chromosomes to opposite ends, nuclei reform, and then the cell splits. Now, a plant cell looks very similar to this, except that, in fact, it looks the same, except that plants don't have centrioles and they have cell walls around them, but it's a very similar looking process. See, there's the same basic processes, except now when the cytokinesis occurs in the plant cell division, the plant cell cannot pinch in. It literally just forms a plate. The plate is formed from the microtubules that were used to separate the chromosomes. And that plate then allows the two cells to be separate. Then each cell builds its own cell membrane and cell wall on its own side, and the division is complete. We have two cells where we had one before. That's mitosis. The only differences between plant and animal cells were pointed out. Plant cells do the cytokinesis differently, and they do have plant cells are missing centrioles that animal cells have. So that's it for mitotic cell division. The next thing we got to talk about is meiotic cell division.